let's talk about some synthetic antimicrobial drugs. So these are chemicals that can be consumed by humans to combat pathogenic microbes. Now, they might not only target pathogenic microbes, they might target all microbes in the body, but they will not target and should not harm human cells, at least not in the same way that they are harming the microbial cells. So again, these are all synthetic. They were discovered and designed in a lab. And these are analogs. So what is an analog? An analog is something that has a similar shape and usually, or not always, binding capacity to a naturally existing molecule. So this means that we are talking about molecules that look similar and usually have a very similar composition to molecules that naturally exist. And by using these kind of replacements, they're kind of like a knockoff. So usually you could tell a knockoff purse or a knockoff watch from the real thing because they're very similar. They probably have the same colors and the same pattern, but there's something off about them. There's something different. So you could kind of think of these analogs as knockoffs. So some examples of categories of analogs are growth factor analogs, nucleotide analogs, and quinolones. So growth factor analogs are knockoffs, so to speak, of growth factors. So growth factors are molecules in the environment, and they tell or help microbes to grow. Grow and divide. And so these growth factors are necessary for growing and dividing. They're not just convenient or nice to have, they are completely necessary. And oftentimes growth factors are referred to as essential molecules. So if you're familiar with the nutrition facts on the back of a cereal box or something, sometimes they'll say that this food contains essential amino acids. What are essential amino acids? Well, don't we need all 20 amino acids? Well, yes, but we can make some of them. Our metabolism can produce some of them, but we do not have the enzymes to produce all of the 20 amino acids. So the ones that are essential are those that we cannot make on our own and we need to consume through our diet. And so some growth factors are essential for bacteria, and the bacteria cannot make them. So again, growth factors cannot make these essential growth factors, and so when you introduce an analog of a growth factor, then the bacteria is kind of stuck because it has to have that molecule. It can't make it itself, and so it ends up using these analog versions. Now we know what nucleotides are, and we're going to talk about this more in a minute. But let's go over quinolones. So quinolones are an analog for DNA gyrase. DNA gyrase is a molecule that bacteria do make, but it is crucial for copying DNA because it helps to unwind DNA and prevent the DNA from breaking. So DNA gyrase is for DNA replication. And this is more of a case of introducing this might outcompete the actual DNA gyrase, or at the very least is going to slow the DNA replication process because this analog is not going to perform the same job that the real DNA gyrase would. And so a couple more specifications here, special things about DNA gyrase. So just to be clear, this is an enzyme. And DNA gyrase is also important because it is a gene that is shared amongst almost all bacteria. And so if you're trying to compare all bacteria in some environmental sample, you're going to get tons of different types of bacteria. So how do you compare them? You don't want to sequence all of the DNA of all the bacteria because that's time consuming, it's expensive, and you also then just have a huge amount of data that you need to store somewhere on some lab computer. So one thing that you could do is use something called housekeeping genes. And housekeeping genes are a set of genes which nearly all microbes that we know of have, and they are essential, they are necessary. I'm going to try not to use essential in the colloquial sense because we're talking about essential amino acids and things like that. Housekeeping genes are genes that bacteria absolutely need to have express in order to survive. So these are things like DNA gyrase. Another one would be a gene for other genes for DNA replication and repair. And so there's a gene called Rep-A, which is another housekeeping gene that's sometimes sequenced. And there are also genes for certain pathways, but when we start to talk about specialized pathways, then not all bacteria have them, right? So we have been talking about how different bacteria might grow on different media. Some bacteria will grow towards the surface of a container of broth, while others will grow at the bottom because of their tolerance for oxygen. So those differences that you can observe in the lab are going to be reflected in their genomes. So you wouldn't want to use those types of genes to try and sequence all of the bacteria in your sample. So let us move on and take a closer look at the nucleotide analogs, or at least one nucleotide analog. And we'll talk a little bit more specifically about how analogs function and why they can be really detrimental for bacterial growth. So an analog, I've done a little doodle here, and this is not an analog necessarily, this is just the induced fit model, which we've talked about in the past, induced fit model for enzyme function. And binding. So recall that in the past, the mid-1900s, so like 1950s, 1960s, scientists thought that enzymes never changed shape. They were always the way they were. Now we know that this induced fit model is true for enzymes. Enzymes do change shape while they are doing their job, while they are binding some substrate. So here we see the enzyme and we see a substrate. The enzyme's active site here is this little triangular shape. It doesn't perfectly mirror the substrate shape, right? The substrate is rounded. This is made up of sharp angles. And when they bind, both of them change to become a little bit more similar to the other. And so this is an image or a description of a descriptive image of when they're bound together. We see that the bottom of the enzyme binding site stays straight, but the substrate changes to match that shape. but the top here, the enzyme changes, on the other hand, to match the rounded substrate shape. Now it's not always this equal, but this is a good analogy for how that works. Then the enzyme returns to its original shape, which is why scientists originally thought that enzymes didn't change shape, because they were never able to look at the enzymes like this. They only saw them like this. And the substrate, on the other hand, has permanently changed shape because we have made our product. So I guess a P instead of an S. Okay, so put that aside for a moment, and let's look at these nucleotide analogs. I'm just going to erase this so that this is not so busy. So here on the left side, we see uracil, and on the other side, we have 5-fluorouracil. Now, these two molecules are extremely similar. Uracil is, of course, the naturally occurring nucleotide, which is used in mRNA, and 5-fluorouracil is an analog of uracil. Compare. So we see an oxygen here, we see a nitrogen here to the left of it, then we see another oxygen if we're going around the ring counterclockwise, then we see a nitrogen at the bottom, and then a double bond, and then here is where we have the only difference between these two molecules. On the uracil, we have a small hydrogen atom, whereas on the 5-fluorouracil, we have a fluorine atom. Now, a fluorine atom is much larger than a hydrogen atom, and it's also much more electronegative. So this means that it's not going to act exactly the same as uracil. Again, this is an analog. So what might happen, there's a couple of ways that analogs work. Analogs can sometimes be bound by enzymes, but then you might get stuck in this state so that the enzyme and the analog stay permanently bound because the enzyme is kind of trying to change the analog the way it would change the substrate, but it just can't get that chemical reaction to go forward. The right atoms or the right electrons aren't there, and so it stays stuck, and now there are no enzymes to perform the job that are supposed to be performing this job. And even if you have the original molecule, it's not going to be processed because there's no enzyme to process it. Or you can have an analog that can be bound by the enzyme, and then the analog is not going to perform the same function. It's not going to turn into the same product. And oftentimes we see that analogs, at least the most useful ones, will have a better binding efficiency to enzymes. And what does that mean? It means that the enzyme will selectively bind to the analogs. Now, why does that happen? It's just the chemical structure and the interactions. So it's not that all analogs are naturally better at binding enzymes, it's that we've chosen to use the analogs that are best at it. And that means that these analogs are preferentially going to be associating with whatever enzyme, again, kind of blocking out the naturally occurring molecule from being used and from sometimes carrying out its function. So this, again, is a discussion of analogs and how they work to block microbial growth.